False positive results have always been a challenge in PCR-based genotyping technique, especially when detecting rare variants in a genome. In clinical and research settings, even a single undetected or incorrectly identified allele can significantly affect disease diagnosis and treatment decisions. Therefore, accuracy in distinguishing true variants from background noise is critical. This challenge has led to the development of enhanced amplification strategies, such as RNase H-dependent amplification system, which can be considered a modified form of PCR. It was designed to improve specificity, as primer-dimer formation and non-specific amplification may be there on PCRs. To understand this system, we can compare it to the familiar concept of two-step verification in a security system of TikTok or Facebook. Instead of relying on a single confirmation step, the system uses an additional verification layer to improve reliability. In standard PCR or allele-specific PCR, primers are bound to the target sequence and amplification are proceeded if binding is sufficiently stable. However, in some cases, especially in rare variant detection, mismatch may still give room for compromising by a DNA polymerase and hence allow for amplification and gives the false positive result. Let's understand this concept with very basic simulation that I have prepared, and here is the anatomy of a primer that we will be using. The primer contains DNA primer region, RNA base and blocking region at 3' end. If there is perfect complementation of DNA region along with the RNA with the target base, then the RNA enzyme will recognize this RNA-DNA hybrid and proceed ahead the cleavage of blocking regions, and hence efficient downstream amplification takes place. However, if target contains a mismatch, the hybrid structure becomes less stable and hence enzymatic processing does not occur, and amplification efficiency gradually decreases, reducing the likelihood of false positive signal generation. You can understand this in way that simple way that the enzymatic processing will not occur and the blocking region will stay there, and hence amplification will not take place at that point. Due to its enhanced specificity, it can be used in SNP genotyping, rare variant detection, CRISPR validation, multiplex assay, etc. But can be expensive, so it must be used based on the context and goal of our research.